Good afternoon to all. In last class, I have discussed about cholinergic transmission. So in that, I have discussed how acetylcholine is synthesized in nerve terminals and how it is released from the nerve terminals and how it is deactivated in the synaptic left region. Okay. So in today's class, I'm going to discuss about cholinergic receptors and cholinergic drugs. Okay, so classification of cholinergic drugs and the types of cholinergic receptors and how that cholinergic drugs are binds with the cholinergic receptors and how that cholinergic drugs binds with cholinergic receptors and how acetylcholine will alter the biological responses of the organs. Okay, that things we will discuss now. First thing is cholinergic drugs. Okay, classification of cholinergic drugs. So cholinergic drugs, also known as cholinomimetic drugs, also known as parasympathomimetic drugs. Okay, so cholinergic drugs, because of acetylcholine, it is a major neurotransmitter in parasympathetic nervous system. Okay, so cholinergic drugs is a major cholinergic drugs is also known as what? Cholinomimetic drugs and also known as parasympathomimetic drugs. And cholinergic drugs is divided to two categories, that is cholinesters and alkaloids. Okay, so cholinesters examples is acetylcholine, methacholine and carbachol. Okay, and bethanychol. And alkaloids examples are muscarine, pilocarpine and aerocholine. Okay, so acetylcholine is binds with two kinds of receptors. So that is muscarinic receptors and nicotinic receptors. Okay, cholinergic drugs are binds with two kinds of receptor. Either it is muscarinic or nicotinic receptors. If acetylcholine nothing but cholinergic drugs, if it is binds with the muscarinic receptors, means it produces the muscarinic like action. Okay, if it is binds with the nicotinic receptors, means it produces a what kind of actions? That is nicotinic like actions it will be produced. So depending upon the type of receptor binding, that acetylcholine is produces the either muscarinic action or nicotinic action. Okay, so next thing is the cholinergic drugs is binds with two kinds of receptors, that is muscarinic receptor and nicotinic receptor. So this slide is I have already discussed in last class also. Muscarinic receptors is also known as what? G protein coupled receptors. Nicotinic receptors is also known as what? Ligand gated cation channel. Okay, and what is the agonist for this receptor is muscarine, and nicotinic receptor agonist is nicotine, and antagonistic for muscarinic receptor is atropine, and antagonistic for nicotinic receptor is D-tuberculin. And the muscarinic receptors is divided into 5 major types. Okay, so in that M1, M2, M3, M4, M5. Okay, so in this M1, M2, M3, first 3 muscarinic receptors are major receptors. Why? Because this M1, M2, M3 is present in both peripheral organs and central organs. Okay, present in central region and peripheral organs. But M4 and M5, M4 and M5, these two receptors are present in only central region. Okay, but M1, M2, M3 is present in both what? Central region and peripheral region. Okay, so that's why first 3 M1, M2, M3 are major muscarinic receptors. Okay, and functionally M1, M3, M5, M1, M3, M1, M3, M5 are excitatory receptors. Okay, and mechanism of action is also similar, M1, M3, M5, and M2 and M4 is the inhibitory like receptors. Okay, and nicotinic receptors are two types, that is nicotine type of nicotine receptor and muscarinic type of nicotinic receptors, and these are usually excitatory receptors. Okay, and nicotinic receptors are excitatory receptors. And next thing is, so today I will discuss about M1 receptor and M3 receptor. Okay, M1 receptor and M3 receptors are M1, M2, M3 is a major receptor because they are present in both central region and peripheral region. Okay, and M1 and M3 both are M1, M3, M5. Okay, there comes under one category. Functionally wise, M1, M3, M5 is comes under one category, is an excitatory receptor. Okay, so the mechanism of action of M1, M3, M5 is same. Okay, and first M1, M3 I will discuss now. The thing is, what is the location and what is the function. Okay, what is the location of M1 receptor, M3 receptor, and what is the function of that receptors, and what is the nature of the receptor, and what is the mechanism of the receptor, and what is the agonist and antagonistics of that receptors. Okay, so first one is the location of M1 receptor is autonomic ganglion. Okay, and gastric glands also, and CNS region. Okay, so a function is depolarization effect, and acetylcholine action gastric glands is increases the gastric acid secretion. Why? Because acetylcholine is stimulate the release of histamine. Okay, actually histamine is involved in allergic responses, but histamine action on gastric glands is what increases the secretion. Why? Because gastric glands consist of histamine receptors. Okay, so our cholinergic drugs are stimulate the what histamine release. Okay, so histamine is binds with histamine receptor on gastric glands. So then what will happen? It stimulate the gastric acid secretions. And CNS region also. Okay, function is involved in learning process, memory, and motor functions also. And M3. Okay, so M3 is present in visceral smooth muscles. Okay, it is present in visceral smooth muscles, and function is what? Contraction effect. Okay, smooth muscle contraction, and it is present in iris region. Okay, and ciliary muscles also, ciliary muscles of eye also. So effect is constriction of pupil and contraction of that ciliary muscle, and it is present in exocrine glands. Okay, M1 receptors are present in gastric glands, and M3 receptors are present in exocrine glands. So it is regulate the what? Sweating and salivation. Okay, so it stimulate the secretions. And M3 receptors is present in vascular endothelium. Endothelium means what? It is the inner layer of blood vessels. Okay, so what is the function is vasodilation. Why? Because M3 receptors is G protein coupled receptor. So here it is a G protein coupled receptor, and the involved mechanism is IP3 and DAG pathway. Okay, so this pathway is releases the somewhat releasing factor, that is endothelial releasing factor, EDRF. Okay, so that endothelial releasing factor is it relaxes the what vascular endothelium. It causes the vasodilation, relaxation of blood vessel. Vasodilation means relaxation. Constriction means contraction of the blood vessels. So here cholinergic drugs act on vascular endothelium is vasodilation. Because of it releases the relaxing factor. The relaxing factor name is EDRF, endothelial relaxing factor. Okay, so that factor will relaxes the blood vessels. So that is called as vasodilation. Okay, and M1, M2, M3. Sorry, M1 and M3. So these three receptors are what kind of receptors? That is G protein coupled receptors. Okay, so the nature is exactly GQ protein coupled receptors. GQ protein coupled receptor. So in the mechanism of action of M1, M3 is similar. That is, it is active. It is act through two kinds of pathways. That is IP3 DAG pathway, same. M3 is also same. IP3 DAG pathway or PL2 pathway, phospholipase A2 pathway. So this in this pathway there is a increase in the synthesis of prostaglandins. Okay, so here M3 is also same. It is act through ML2, PL2, sorry, PL2 pathway, phospholipase A2 pathway. Okay, in this pathway there is an increase in the synthesis of prostaglandins. Okay, so cholinergic drugs effect on gastric glands is increase the secretion. Cholinergic drugs effect on visceral smooth muscle is contraction. How it is? how it is, how acetylcholine is induces the smooth muscle contraction, how cholinergic drugs is induces the contraction of ciliary muscle, how acetylcholine drugs, sorry, nothing but cholinergic drugs is induces the constriction of pupil. Okay, how it is stimulate the exocrine gland secretion, because M1, M3 receptors are G protein coupled receptors, and they are act through two kinds of pathways, IP3DAG and PL2 pathway. So first thing, what is G protein coupled receptor? Okay, so the G protein coupled receptors is also known as transmembrane receptors. Okay, also known as what transmembrane receptors. What is meant by transmembrane? Okay, so transmembrane receptors means the receptors which will be existed across the cell membrane. Okay, so it will be present across the cell membrane, and what it is also known as heptahelical receptors. Okay, and these are the large family of cell membrane receptor. Okay, G protein coupled receptors are large family of cell membrane receptors, and they are associated with what? They are linked with what? Effectors through what? GTP activated proteins. Okay, so this one is GTP activated proteins. GTP means guanosine triphosphate activated proteins. So why this receptor is called as G protein coupled receptor? Because of this receptor is linked with effectors through what? G proteins. So that's why it is called as G protein coupled receptors. G means what? Here the GTP activated proteins. The proteins are activated due to what? GTP, that is guanosine triphosphate. That's why it is called as G protein coupled receptor. And this receptor molecule has a 7 helical membrane spanning hydrophobic amino acid segments. Okay, and they consist of two domains. G protein coupled receptor consists of two domains. See this one is extracellular domain and this one is intracellular domain. And extracellular domain consists of what? That is amine group. Okay, and intracellular molecule consists of carboxylic group. Okay, and this intracellular molecular that's intracellular domain, this one is carboxyl group. Carboxyl group is presented intracellular domain. So this domain is associated with what? GTP proteins. Okay, it is associated with what? GTP proteins. Okay, so these GTP proteins are two types. Okay, in G protein coupled receptor there are two types of proteins is there. Monomeric G proteins, trimeric G proteins. Okay, so here I am discussing trimeric G proteins. Okay, why it is called as trimeric G proteins? Because they consist of three subunits, that is alpha, beta and gamma. Okay, three subunits is there. Okay, that's why it is called as trimeric subunit, trimeric G proteins. Okay, so in resting condition, okay, so in resting condition, the alpha subunit, in resting condition, the alpha subunit is binds with what? GDP, that is guanosine diphosphate. What? Guanosine diphosphate. GDP is binds with alpha subunit of G protein. Okay, in a resting condition. Okay, there is no, if there is no availability of what? Chemical signal. Okay, if there is no availability of chemical signal, means it is in resting state. In the resting state, the GDP is binds with alpha subunit. Okay, but when agonist is binds with the GPCR. Okay, so when agonist, nothing but any other chemical signal, nothing but any acetylcholine or adrenaline, whatever it is, a neurotransmitter or any other chemical signal. Okay, so when that agonist is binds with this GPCR, then what will happen? There is any conformational changes is happens in GPCR. Okay, so that conformational changes that may undergo the what? Conformational changes in the GPCR. When it is conformation changes is happen, when agonist is binds with GPCR, it may undergo the conformational changes in the receptor. So that changes, okay, that changes will transmit the signals to what? G proteins. Okay, that changes will transmit the signals to what? G proteins. Then what will happen? The GTP is binds with alpha subunit. Okay, so the GDP is replaced by GTP. GTP means guanosine triphosphate. Okay, so when agonist binds with the receptor, then conformation changes happens in the receptor. That changes will transmit the signals to what? GTP proteins, G proteins. Then what will happen? The GDP is replaced by GTP. Then afterwards, what? The G alpha subunit, it will be dissociated from the beta and gamma. Okay, so alpha, beta, gamma. Okay, in this the beta and gamma, it is covalently linked with what? Plasma membrane. Beta and gamma is covalently linked with the plasma membrane, and beta and gamma, it forms a dimer. Okay, so when agonist is bind with the receptor, then GDP is replaced by GTP. Then what will happen? That alpha subunit, okay, that alpha subunit, it will be dissociated from the alpha and beta, nothing but dimer. It dissociated from this dimer. Then what will happen? This alpha subunit, it may interact with effectors, and it produces the second messengers like IP3, DAG, and cyclic AMP. Okay, it stimulates the production of what? Second messenger. Then ultimately it may lead to what? The response it will be generated, because of the second messengers are involved in the response. Second messengers are, it produces the biological responses. Okay, so this one is G protein coupled receptor. Okay, so G protein coupled receptors are large family of cell membrane receptor. They are linked with effectors through what? GTP. Why it is called as GTP? Because of the G proteins are activated through what? GTP means. GTP means what? These are the GTP activated proteins. That's why it is called as G proteins. The G proteins, these are the trimeric G protein. Why it is called as trimeric G protein? Because that G protein consists of alpha subunits, beta subunit, and gamma subunit is there. Okay, this is called as trimeric subunit. Okay, so here next thing is again alpha subunit consists of, okay, alpha subunit consists of again three major proteins. Alpha subunit consists of three major proteins, that is GS, GI, GQ. Okay, so GS means what? That is stimulatory G protein. So this stimulatory G protein, the GS protein is activate the which enzyme? Stimulate the what enzyme? Adenyl cyclase enzyme. Okay, so this adenyl cyclase enzyme is involved in the production of the second messenger. The name of the second messenger is cyclic AMP, means adenosine monophosphate. AMP means adenosine monophosphate. So these three proteins, the three proteins is present in which subunit? Alpha subunit. Okay, so when agonist binds with the GPCR, then immediately GDP is replaced by GTP. Then what will happen? This alpha subunit is dissociated from this dimer, beta and gamma dimer. Then the GS, if GS protein is activated means, then what will happen? It stimulate the adenyl cyclase enzyme, and it produces the second messenger. The name of the second messenger is cyclic AMP. If GI protein is activated means, then what will happen? If GI protein is activated means what? It is inhibiting the which enzyme? Adenyl cyclase enzyme. Okay, it's an inhibitory effect. So inhibiting the which enzyme? Adenyl cyclase. Then inhibiting the which secondary messenger production? That is cyclic AMP. So decrease the cyclic AMP. Cyclic AMP is nothing but secondary messenger. So that messenger production, it will be decreases if GI protein is activated means. Okay, if GQ protein is activated means, then phospholipase enzyme is activated. Okay, phospholipase C enzyme is activated. Then it produces the second messenger. The name of the second messenger is IP3, DAG, and calcium. Okay, so here it is our receptors M1 and M3 receptors. Okay, M1 and M3 receptors are, the nature of the M1 and M3 receptor is GQ protein coupled receptor. Both muscarinic receptors are G protein coupled receptor. Again, M1 and M3. Okay, M1, but M1 and M3 is GQ. GQ means the subtype. Okay, the G protein consists of 3 subunits, alpha, beta, gamma. Again, alpha consists of 3 subunits. Okay, that is what? G, S, GI and GQ. Okay, so in this M1 and M3. Okay, M1 and M3 nature is GQ protein coupled receptors. So then which enzyme is activated? Then if cholinergic drugs is binds with muscarinic receptor 1 and 3, then what will happen? Which enzyme is activated? That is phospholipase enzyme C is activated. Then which secondary messenger is production is happens? That is IP3 and DAG. Okay, so then what will happen? Then phospholipase C pathway, because of M1 and M3 is the nature is GQ protein activated, GQ coupled receptors. Okay, so here for example here histamine is there. Instead of histamine, acetylcholine is also stimulate the release of histamine. Okay, so and cholinergic drugs. Okay, another cholinergic drugs is binds with the respective receptors, that is G protein coupled receptors. Then what will happen here? Histamine is also, histamine receptors is also, the nature of the histamine receptor is also G protein coupled receptors. Okay, after signal, okay, nothing but agonist is binds with receptors. For example, cholinergic drugs is binds with the receptors, cholinergic receptors. Okay, the cholinergic receptors type is especially muscarinic type. I'm discussing here M1 and M3 receptors. M1 M3 receptor mechanism. Okay, so after binding with the receptor, then what will happen? This receptor is associated with what? G proteins. Here, which protein subunit is activated? That is GQ. Okay, alpha, beta, gamma, 3 subunits is there. Alpha consists of again GQ subunit is there, and the GQ protein is activated. So then what will happen here? It is the GDP is removed. Okay, in resting state, GDP is binds with GQ protein, but after binding with after binding of signal with receptors, then in this receptor, then what will happen? There is a conformational changes happens. Then the change will transmit the signals to what? G proteins. So then what will happen? Instead of GDP, the GTP is binds with the GQ proteins. Okay. Then after binding of GTP with the GQ proteins, then what will happen? Then immediately it is dissociated from this dimer, beta and gamma. Okay, after dissociation, see this. Okay, GQ, this alpha subunit consists of again subtype is GQ, and the GQ alpha subunit of GQ protein, it binds with what? GTP. Okay, so then what will happen? After dissociation, this GQ protein is activates the phospholipase enzyme C. PLC means phospholipase enzyme C. Then phospholipase enzyme C, phospholipase C, it is hydrolyzes the PIP2. So what is PIP2? PIP2 means, note down, full form of PIP2 means phosphatidyl inositol 4,5 bisphosphate. Okay, phosphatidyl inositol 4,5 bisphosphate. Okay, so what is after activation of PLC? The PLC phospholipase C, it is hydrolyzes the PIP2. Okay, so normally this PIP2 is present in cell membrane. Okay, it is present in cell membrane. So after hydrolysis of PIP2, upon the hydrolysis process, then here it is 2 kinds of messengers it will be activated. So that is DAG messenger is activated, and IP3 messenger it will be activated. Okay, so means activity is nothing but the production of the DAG and IP3. production is occurs IP3 and DAG. Why it is? Why? Because of the PLC is hydrolyzes the PIP2. PIP2 is present in what? Cell membrane. PIP2 means what? Phosphatidyl inositol 4,5 bis phosphate. Okay. Upon hydrolysis process, two kinds of messengers it will be produced. That is DAG and IP3. Then what will happen? This IP3 is stimulate the release of calcium from the endoplasmic reticulum. Okay. So what is endoplasmic reticulum? Endoplasmic reticulum, it is a major storage site of calcium also. Okay. Endoplasmic reticulum, it is a storage site of calcium also. So this IP3 is what IP3 is generates a calcium. Okay, it stimulates the release of what? Calcium from the endoplasmic reticulum. See this one is endoplasmic reticulum. IP3 stimulates the release of calcium from the endoplasmic reticulum. Then this calcium, okay, so this calcium is entered into the cytosol. Okay, at the same time, the DAG means, note down, DAG means diacyl glycerol. Okay, so this DAG with the help of this cytosolic calcium, see this, the plus mark is there, DAG with the help of calcium, cytosolic calcium, then protein kinase C. Okay, the protein PKC means protein kinase C enzyme is activated. Okay, with the help of calcium, the DAG is activated PKC with the help of calcium. Okay, protein kinase. What is the purpose of protein kinase? Protein kinase, it is involved in phosphorylation, protein phosphorylation. Okay, what kind of proteins? That is structural proteins and functional proteins. Okay, so PKC it is activate the both, phosphorylate the both structural proteins and functional proteins. Okay, at the same time, this calcium, it binds with CAM. CAM means what? Calmodulin. CAM means calmodulin. What is calmodulin? Calmodulin is nothing but it is a calcium binding protein. Okay, what is CAM? CAM is nothing but calcium binding protein. Okay, so this calcium is binds with calmodulin. Then afterwards what will happen? It is activate the MLCK. What is MLCK? That is myosin light chain kinase. MLCK means myosin light chain kinase. So what is myosin light chain kinase? So myosin, actin, okay, these are the proteins. Okay, so these proteins are involved in muscle contraction. Okay, so MLCK, the myosin light chain kinase, okay, this is an enzyme, it is involved in muscle contraction. Okay, so what is the cholinergic drugs effect on visceral smooth muscles? That is visceral smooth muscle. Visceral smooth muscles consist of what kind of receptors? Watch. Visceral smooth muscle consists of what kind of receptor? That is M3 receptors. Okay, and iris ciliary muscles, exocrine glands consist of M3 receptors. Okay, so here what is the action? What is the action? That is calmodulin, after binding with cytosolic calcium, it is activates what? Myosin light chain kinase. Okay, actin and myosin, these are the proteins. These proteins are involved in the what? Muscle contraction. Okay, so that's why what is the action of acetylcholine on visceral smooth muscle is contraction. And how that contraction effect it will be produced? Because after signal binds with the receptor, then what will happen? GQ protein is activated and it is dissociated from this dimer. Then it is activate the phospholipase C. Then it is hydrolyze the PAP2. Then production of the two kinds of messengers. So that is secondary messengers. That is IP3. What is IP3? Inositol 1,4,5 triphosphate. Okay, so then DAG means what? Diacyl glycerol. So IP3 stimulates the release of calcium from the endoplasmic reticulum. Then calcium is reactive with what? Calmodulin. It activates the myosin light chain kinase. Okay, they are involved in the muscle contraction. At the same time, Calcium is also binds with calmodulin. It is activate the myosin light chain kinase and calcium calmodulin protein kinase. CCPK means calcium calmodulin protein kinase. So what is the function of this one? CCPK, it is phosphorylate the what? Another CCPK is phosphorylate the another proteins. Okay, those proteins which are involved in the muscle contraction. So that protein phosphorylation is occurs by CCPK. Okay, and what after the cytosolic calcium, it again it is recycled by uptake into what? Uptake into endoplasmic reticulum with the help of pump. Okay, as well as it will be efflexed. What it is efflexed to outside, means it is efflexed by which membrane? That is calcium pump. It is present in cell membrane. Okay, so it is recycled by it is uptake into sacroplasmic reticulum, means uptake into sacroplasmic reticulum, as well as it will be efflexed by membrane calcium pump. Okay, then what if this termination, if agonist, okay, so this pathway is degraded by termination of the second messenger. Okay, this pathway is degraded by termination of the second messenger. So what is that? IP3 is dephosphorylated to inositol. IP3 is dephosphorylated to inositol. What is inositol? Inositol, it is again what is inositol? Again, it is reutilized in the synthesis of PIP2. Okay, and again the DAG is partially converted back into phospholipids. Okay, DAG is partially converted back into phospholipids, and the IP3 is dephosphorylation is occurs. Dephosphorylation of IP3. Then the remaining product is inositol. Inositol is required for the resynthesis of PIP2. Okay, so the termination of this action is occurs due to decrease in the production of the second messenger and decrease in the activation of the PLC. If this decrease in the activation of PLC means there is no hydrolysis of PIP2 does not occurs. Okay, so this one is IP3 and DAG pathway. Okay, so in this pathway mainly the contraction effect will be produced. How it? Because what is the cholinergic drugs? Cholinergic drugs effect on visceral smooth muscles is contraction effect, and what is the acetylcholine action on gastric glands is it stimulate the gastric secretion. Okay, because of this pathway only, and the contraction of what pupil and the contraction of ciliary muscles also. So how it is occurs? Because this pathway. Okay, and next thing is PLA2 pathway. So what is PLA2 pathway? See this. So M1 and M3 receptors, it produces a contraction effect in visceral smooth muscles, and gastric gland secretions also, exocrine gland secretion and gastric gland secretions is also occurs. So it is act through two kinds of what pathways? IP3 and DAG pathway and PLA2 pathway. Okay, so next class I will discuss about PLA2 pathway. Okay, PLA2 pathway and what is the agonist and antagonistics of this pathway, and M2 receptors. Okay, so what is that M2 receptor? M2 receptors location and function and the nature of that receptor, and cyclic AMP pathway. It is acted through cyclic AMP pathway, M2 receptors. And next class I will discuss about PL2 pathway and M2 receptors structure and location and function and cyclic AMP pathway. Okay.